Cut and run is a versatile in vivo method used to map protein-DNA interactions. This video will provide a detailed visual demonstration to guide your cut and run experiments. This cut and run protocol has been optimized by scientists at Cell Signaling Technology for use with the cut and run assay kit. The full protocol is available on cellsignal.com and linked in the description below this video. You can also find answers to frequently asked questions, a troubleshooting guide, links to technical support, and recommended companion products and validated antibodies at the CST Cut and Run Resource Center. Visit anytime using the links below this video. The cut and run assay relies on the specific binding of an antibody to a protein of interest, which may be a histone modification, transcription factor, or cofactor. The enzyme protein A protein G micrococcal nuclease is added next and is recruited to chromatin by the interaction of the antibody with protein A or protein G. When calcium is added, the enzyme cuts the DNA at sites of the protein-DNA interaction. The complex containing the digested DNA is free to diffuse into the supernatant, while remaining chromatin remains trapped in the nuclei, simplifying subsequent DNA purification. Cut and run antibodies are thoroughly validated in-house by scientists at Cell Signaling Technology using the same cut and run protocol, cut and run assay kit, and companion reagents featured in this video. The cut and run protocol is divided into sections, and this video is organized into corresponding chapters which can be accessed using the timeline at the bottom of this video. We recommend that first-time cut and run users make sure to watch the Protocol Considerations chapter for guidance on choosing protocol variations and designing your experiment. The first 6 sections, from the start to purified cut and run DNA, can be performed in as little as 1 or 2 days. The CST Cut and Run protocol includes several safe stop points where samples may be stored before resuming the protocol. Safe stop points are noted in the video and in the full protocol and give the user flexibility for planning the workflow. Cut and Run DNA is compatible with analysis by both quantitative PCR, or qPCR, and next-generation sequencing, abbreviated as NGS. For researchers who use NGS analysis, CST offers a separate kit for preparation of cut and run DNA libraries. After you have completed the cut and run protocol and verified it is working by qPCR, please refer to the protocol for the DNA library prep kit for Illumina systems, ChIP-seq Cut and Run, which can be found in the CST Cut and Run Resource Center. You may also watch the accompanying protocol video for DNA library prep for cut and run by clicking the pop-up link in the corner. Before setting up your cut and run experiments, let's review several important aspects of the cut and run protocol that can impact your results. Consider the number of cells required to generate robust cut and run results, potential sensitivity of your cells to digitonin, whether to use live or fixed cells, or if using tissue, what protocol variations might be required. First, consider your sample type and target protein when determining the number of cells required for input samples and when choosing which protocol variation to use for Section 1, Cell and Tissue Preparation.
One cut and run reaction can use 5,000 to 250,000 cells, or 1 to 5 milligrams of tissue. Whenever possible, we recommend using 100,000 cells or 1 milligram of tissues as a starting point. If cells are limiting, we recommend using at least 5 to 10,000 cells per action for targets that are histone modifications, and 10 to 20,000 cells for targets that are transcription factor or cofactors. In cut and run, digitonin is used to permeabilize cells, allowing the enzyme and the antibody to access the chromatin. The amount of digitonin in this protocol is sufficient to permeabilize most cell lines. If you are concerned that your cell or tissue types aren't sufficiently permeabilized, refer to Appendix A of the full cut and run protocol to quantify permeabilization efficiency with trypan blue. If less than 90% of your cells are stained by trypan blue, consider titrating with varied digitonin concentrations. Once you have identified a digitonin concentration that permeabilizes at least 90% of your cells, apply it to the working buffers used in the protocol sections 2 through 4. When deciding between live versus fixed cell protocol variations, we advise using live cells whenever possible. If these are not accessible, or if the live cell preparation method in Section 1A fails to generate robust cut-and-run results, consider switching to the fixed cell preparation protocol in Section 1B, which uses a relatively light fixation method of 0.1% formaldehyde for 2 minutes. This protocol variation should also be considered if your cell pellets need to be frozen prior to beginning of the experiment, or if your cells are fragile and prone to lysis, or if the signaling pathway in your cells are sensitive to binding by contaminated beads. T cells, NK cells, and other immune cells can benefit from light fixation. Finally, fixation may also help to boost enrichment of low abundance or weakly binding protein targets for all cell types. For tissue sample preparation in Section 1C, consider the target protein and the tissue type when deciding if fixation is needed. Formaldehyde fixation is not required for enrichment for histone modifications. However, the enrichment of many transcription factors and cofactors from tissue does require a light fixation step with 0.1% formaldehyde for 2 minutes for optimal results. We recommend this approach for most tissue types and protein types. A medium fixation step with 0.1% formaldehyde for 10 minutes may improve results for lower abundance and/or weak binding transcription factors and cofactors. It may also improve results for difficult tissue types such as fibrous tissues. Note that overfixation of cells or tissue should be avoided, as this can inhibit the cut-and-run reaction.
Now, let's take a look at some of the other components of a cut-and-run experiment that can help you interpret your data. For both qPCR and NGS analysis, we recommend including cut-and-run validated positive and negative control antibodies to show that your cut-and-run assay is working. Both the trimethylhistone H3 positive control antibody and rabbit IgG isotype control antibody shown here are included in the cut-and-run assay kit, along with relevant primer sets for loci with known binding characteristics.
For design of controls, we strongly recommend using the negative control antibody and not a no antibody control, because the latter results in high level of non-specific micronucleus digestion and high background signal. When designing experiments with NGS analysis as an endpoint, we recommend performing real-time qPCR first as a quality control step. Based on your qPCR results, you can either confirm that the cut-and-run protocol is working well in your hands or decide to troubleshoot your experiment when needed, avoiding the lost time and cost spent on unnecessary sequencing. We recommend using the input sample DNA as a negative control in both qPCR and NGS analyses. Input DNA is also used for calculation of the signal-to-noise ratio for NGS.
This flowchart illustrates options for preparation of input DNA. For NGS analysis, fragmentation of the input DNA is required. Section 5 of the cut-and-run protocol includes steps for preparation of input DNA using sonication and is featured later in this video. If a sonicator is not available, an alternate protocol for enzymatic digestion of cut-and-run input DNA can be found linked on the cut-and-run resource page.
If performing qPCR only, fragmentation of input DNA is not required. However, unfragmented input DNA should be purified with phenylchloroform extraction and ethanol precipitation. Many users prefer instead to fragment the input DNA and purify it with spin columns because this protocol is simpler and faster. For NGS controls, input DNA fragmented either by sonication or enzymatic digestion is preferred over IgG. Normal IgG antibodies generate low-diversity DNA libraries and can generate non-specific enrichment of genomic regions in cut-and-run. Overall, input DNA is a better indicator of whether your target antibody is working well in assay.
The cut-and-run method results in low levels of background DNA. Heterologous spike-in DNA may optionally be added after digestion to facilitate accurate quantitation and normalization from sample to sample and from experiment to experiment for either qPCR or NGS analysis. When planning your experiment, account for all experimental samples you will need, plus input samples, positive antibody, and negative antibody controls. Note that if you will be performing spike-in normalization for both qPCR and NGS, you should plan to prepare duplicate samples, as these methods require different quantities of spike-in DNA. In each protocol section, scale the volume of buffer being prepared based on the total number of reactions you will be performing. The cut-and-run assay kit includes the components listed here. Add a cut-and-run validated antibody to your target of interest. If one is not available, please refer to the frequently asked questions, linked in the cut-and-run resource page, for guidance on choosing an antibody. If you will be performing fixed cell or tissue preparation, you will also need the items shown here. DNA purification buffers and spin columns for use in Section 6 of the protocol are not included in the cut-and-run assay kit and are available separately. You will also need the following equipment: a magnetic rack that can work with volumes as low as 50 microliters, a refrigerated or cold room microcentrifuge, a lab freezer, a lab refrigerator or cold room with a rotator or rocker for small tubes, a PCR thermocycler, a heat block, an incubator with shaking, and a probe tip sonicator for fragmentation of input sample chromatin, if following the recommended procedure in Section 5. We recommend optimizing sonication conditions for your sonicator model. SafeLock tubes are recommended for storing input samples prepared in Section 1, because they will be heated to 55 degrees in Section 5. Alternatively, screw cap tubes or cap lid locks may be used. In Section 1, cells or tissues are harvested and permeabilized using digitonin. Samples collected are used for cut-and-run reactions and for input DNA controls. If you haven't already, please review the sample and target considerations introduced in the Protocol Considerations chapter of this video before choosing a protocol variation. You can skip ahead or jump back in the video to follow your chosen protocol variation. We will begin with live cell preparation, Protocol Section 1A.
For live cell preparation, we recommend preparing concatenated beads before preparing the cells. This minimizes the time your live cells are waiting before being combined with the beads. This video follows the recommended order of steps, starting with Section 2, steps 1 through 5, then proceeds with the steps for live cell preparation.
Before starting live cell prep, thaw digitonin solution at 90 to 100 degrees. Thaw spermidine and protease inhibitor at room temperature. Once thawed, keep the protease inhibitor stock at room temperature, not on ice. Inspect the digitonin solution to check it is completely thawed, then move to ice as it will be needed later in the protocol. Place the concanavalin A bead activation buffer on ice. For each reaction, prepare 100 microliters of antibody-binding buffer with spermidine, protease inhibitor cocktail, and digitonin solution, as indicated in Section 2 of the protocol. Mix by pipetting up and down and place on ice. Begin bead preparation by carefully resuspending the stock of concanavalin A magnetic beads by gently pipetting up and down. Transfer 10 microliters of bead suspension for each cut-and-run reaction to a new microcentrifuge tube. Add 100 microliters concanavalin A bead activation buffer per 10 microliters of beads. Gently mix by pipetting up and down. Place the tube on a magnetic rack until the solution becomes clear. This takes about 30 seconds to 2 minutes. With the tube still on the rack, gently remove supernatant using a pipette. Do not aspirate with vacuum. Remove tubes from the rack and wash beads a second time by repeating steps 2 and 3. Add 10 microliters of concanavalin A bead activation buffer per reaction and gently resuspend by pipetting up and down. Store the activated beads on ice during the next steps. We will now proceed with preparation of live cell samples in Protocol Section 1A. Prepare 1x wash buffer with spermidine and protease inhibitor cocktail as indicated in the protocol. For example, for 5 samples, prepare a final volume of 2.5 milliliters by combining the components in this table. Equilibrate 1x wash buffer to room temperature before proceeding to sample collection. Visually inspect the overall health of your cells and density or confluency of suspension and adherent cell lines, respectively. Collect 5,000 to 100,000 cells for each cut-and-run reaction, input sample, or positive or negative control needed. Use an automated cell counter or a hemocytometer to ensure the appropriate number of viable cells are collected. To minimize potential loss of cells, collect each experimental sample in one tube. These will be split into separate tubes for each cut-and-run reaction in Section 2, Step 10. Adherent cell lines need to be detached from the dish before measuring viability and cell count. First, rinse the dish with 1x PBS. Then add trypsin and incubate at 37 degrees to detach cells from the dish, followed by neutralization with 3 volumes or more of culture media. Use of a cell scraper is not recommended since this could disrupt cell and nuclear integrity, affect signaling pathways, or disturb dynamic protein-DNA interactions and impact your cut-and-run results. Before proceeding, consider the number of cells in your sample. We will first demonstrate protocol steps 2 through 6 following the main protocol for 100,000 cells or more, and then discuss necessary protocol variations for low cell numbers. Centrifuge cells for 3 minutes at room temperature. Then remove supernatant media. Resuspend the cell pellet in 1x wash buffer by gently pipetting up and down. Centrifuge the resuspended cells for 3 minutes and remove the supernatant. Wash the pellet a second time by repeating steps 3 and 4. Resuspend in 100 microliters of 1x wash buffer per sample and gently pipette up and down. We will now switch to the low cell number scenario, starting from Step 2.
While working with less than 100,000 cells, the cell pellet is not always visible by eye, and is easy to lose during wash steps. In this case, we recommend leaving about 40 microliters or less while removing liquid from the pellet, and skipping wash steps 3 through 5 in this section.
Centrifuge the low cell number sample from Step 2 for 3 minutes and remove supernatant, leaving 40 microliters or less of liquid. Skip wash steps 3 through 5. Add enough 1x wash buffer to the samples from Step 2 to bring the final volume to 100 microliters per sample and resuspend. For both high and low cell numbers, reserve 100 microliters as your input sample. Transfer to a safe lock or screw cap tube and store at 4 degrees until Section 5. Immediately proceed to Section 2, Step 6. However, if this is your first time watching this protocol video, or if you are concerned about the possibility of clumped beads, please review our tips for handling beads at the start of Section 2 before moving on to Step 6. This section demonstrates the fixed cell preparation protocol variation. Additional materials required for this protocol variation are listed here. Refer to the Materials Safety Data Sheet and guidelines from your local lab safety office when handling formaldehyde. Perform formaldehyde fixation steps 2 through 4 in the fume hood, as demonstrated in this video section. Before beginning fixed cell prep, thaw spermidine and protease inhibitor at room temperature. Once thawed, keep the protease inhibitor stuck at room temperature, not on ice. Prepare 1x wash buffer with spermidine and protease inhibitor cocktail as indicated in the protocol. For example, for 5 samples, prepare a final volume of 2.5 milliliters by combining the components in this table. Equilibrate 1x wash buffer to room temperature before proceeding to sample collection. Visually inspect the overall health of your cells and density or confluency of suspension and adherent cell lines, respectively. Collect 5,000 to 100,000 cells for each cut-and-run reaction, input sample, or positive or negative control needed. Use an automated cell counter or a hemocytometer to ensure the appropriate number of viable cells are collected. To minimize potential loss of cells, collect each experimental sample in one tube. These will be split into separate tubes for each cut-and-run reaction in Section 2, Step 10. Adherent cell lines need to be detached from the dish before measuring viability and cell count. First, rinse the dish with 1x PBS. Then add trypsin and incubate at 37 degrees to detach cells from the dish, followed by neutralization with 3 volumes or more of culture media. Use of a cell scraper is not recommended since this could disrupt cell and nuclear integrity, affect signaling pathways, or disturb dynamic protein-DNA interactions and impact your cut-and-run results. In the fume hood, add an appropriate volume of formaldehyde to each cell suspension to achieve a final concentration of 0.1% formaldehyde. Pipette up and down or invert the tube to mix and incubate at room temperature for 2 minutes. Stop cross-linking by adding the 10x glycine. Pipette up and down or invert the tube to mix and incubate at room temperature for 5 minutes. Before proceeding, consider the number of cells in your sample. We will first demonstrate protocol steps 4 through 8 following the main protocol for 100,000 cells or more, and then discuss necessary protocol variations for low cell numbers. Centrifuge the cell suspension for 3 minutes at 3000 Gs at 4 degrees. Then, remove the supernatant containing fixative in the fume hood. At this stage, you may either proceed directly to step 5, or the fixed cell pellets may be stored at -80, provided you are not working with low cell numbers. Resuspend the cell pellet in 1x wash buffer and gently pipette up and down. Centrifuge the cell suspension for 3 minutes at 3000 Gs at 4 degrees and remove the supernatant. Wash the pellet a second time by repeating steps 5 and 6 of this section. Resuspend in 100 microliters of 1x wash buffer per reaction and gently pipette up and down. We will now switch to the low cell number scenario, starting from step 4.
While working with less than 100,000 fixed cells, the cell pellet is not always visible by eye and is easy to lose during wash steps. In this case, we do not recommend storing cell pellets at -80. Additionally, we recommend leaving about 40 microliters or less in the tube while removing liquid from the pellet, and skipping wash steps 5 through 7 in this section.
Centrifuge the low cell number fixed cell suspension from step 2 for 3 minutes at 3000 Gs and 4 degrees. In the fume hood, remove the supernatant containing fixative, but leave 40 microliters or less of liquid in the tube. Skip wash steps 5 through 7. Add enough 1x wash buffer to the samples from step 4 to bring the final volume to 100 microliters per sample and resuspend. For both high and low cell numbers, reserve 100 microliters as your input sample, transfer to a SafeLock tube, and store at 4 degrees until section 5, and immediately proceed with the rest of your samples to section 2. This section of the video covers tissue sample preparation using light fixation, which is recommended for most tissues and protein targets. Additional materials required for fixed tissue preparation are listed here. Refer to the Material Safety Datasheet and guidelines from your local lab safety office when handling formaldehyde. Prepare the fixation buffer and perform formaldehyde fixation steps 3 through 7 in the fume hood as demonstrated in this video section.
If your experiment requires fresh, unfixed tissue input samples, we recommend preparing concanavalin beads first prior to tissue sample preparation. Refer to the written protocol for guidance.
Before beginning tissue prep, thaw stock solutions and prepare 1x wash buffer with spermidine and protease inhibitor cocktail as indicated in the protocol. For example, for 5 samples, prepare 3.5 milliliters 1x wash buffer by combining the components in this table. Equilibrate 1x wash buffer to room temperature before proceeding to tissue sample collection. Add protease inhibitor cocktail to PBS, mix by pipetting, and place on ice. In the fume hood, prepare 1 ml fixation buffer for each tissue sample. Weigh 1 milligram fresh tissue for each cut-and-run reaction, positive or negative control, or input sample.
For some difficult tissue types such as fibrous tissue, or for low abundance or weakly binding transcription factors or cofactors, the amount of tissue may be increased to up to 5 milligrams of tissue per reaction without scaling up reagents. Additionally, extending the fixation in step 4 from 2 to 10 minutes may improve results for these tissues or targets.
Place tissue sample in a dish and finely mince using a clean scalpel, dissecting scissors, or razor blade. Keeping the dish on ice and keeping the sample cold is important to reduce protein degradation during this step. If using fresh, unfixed tissue, skip to step 9 directly. Otherwise, proceed to fixation. In the fume hood, transfer the minced tissue to a tube with 1 ml fixative solution and swirl to mix. This volume is sufficient for up to 50 milligrams of tissue. For processing larger tissue samples, refer to the written protocol. Incubate tissue in fixative at room temperature for 2 minutes. Stop fixation by adding 10x glycine, invert tube to mix, and incubate at room temperature for 5 minutes. Centrifuge for 5 minutes at 2000 Gs at 4 degrees. In the fume hood, remove supernatant and resuspend the tissue in PBS with protease inhibitor. Centrifuge once more for 5 minutes at 2000 Gs at 4 degrees and remove the supernatant. At this point, you may either proceed to the next step or store fixed tissue pellets at -80. Before resuspending the tissue, pre-cool the Downs homogenizer by placing on ice and cut the end of a 1 ml pipette tip with scissors. Then, resuspend tissue in wash buffer with additives and transfer the Downs homogenizer. Disaggregate tissue into a single-cell suspension by grinding with the pestle. Use 20 to 25 strokes until no tissue chunks are observed. Transfer the homogenized cell suspension to a 1.5 ml tube and centrifuge for 3 minutes at 3000 Gs at room temperature. Remove supernatant and resuspend the pellet in wash buffer. Then centrifuge for 3 minutes at 3000 Gs and remove the supernatant. Wash the cell pellet a second time by repeating steps 12 and 13. Resuspend in 100 microliters of 1x wash buffer per sample and gently pipette up and down. Reserve 100 microliters as your input sample. Transfer to a SafeLock tube and store at 4 degrees until section 5 and immediately proceed with the rest of your samples to section 2. In section 2, magnetic concanavalin A beads bind to the permeabilized cells to anchor them and enable separation. Then, a cut-and-run antibody is added, which binds to the protein of interest located on chromatin.
Concanavalin A magnetic beads simplify cut-and-run sample handling during incubations and wash steps. Researchers are sometimes concerned about the potential loss of beads and sample material when beads clump together or adhere to the walls of tubes. To help reduce clumping and minimize loss of beads, you may consider moving the incubation steps from a rotator to a gentle rocking mechanism or a stationary rack. A quick spin step helps to collect material. We also recommend removing supernatant by pipetting rather than aspirating with a vacuum. Finally, bead suspensions should be mixed or resuspended by pipetting and not by vortexing. Cell lysis, rather than just permeabilization, may contribute to the beads clumping. Make sure your cells are as healthy as possible before beginning the protocol. Incubating cells with beads no longer than 5 minutes in step 7 and treating samples gently during wash steps may also reduce beads clumping. If you follow these best practices and still observe beads clumping, be assured that in our internal testing, beads clumping does not negatively impact cut-and-run results.
Before starting bead preparation, thaw digitonin solution at 90 to 100 degrees Celsius. Thaw spermidine and protease inhibitor at room temperature. Once thawed, keep the protease inhibitor stock at room temperature, not on ice. Inspect the digitonin solution to check it is completely thawed, then move to ice as it will be needed later in the protocol. Place the concanavalin A bead activation buffer on ice. For each reaction, prepare 100 microliters of antibody-binding buffer with spermidine, protease inhibitor cocktail, and digitonin solution, as indicated in Section 2 of the protocol. Mix by pipetting up and down and place on ice. Begin bead preparation by carefully resuspending the stock of concanavalin A magnetic beads by gently pipetting up and down. Transfer 10 microliters of bead suspension for each cut-and-run reaction to a new microcentrifuge tube. Add 100 microliters concanavalin A bead activation buffer per 10 microliters of beads. Gently mix by pipetting up and down. Place the tube on a magnetic rack until the solution becomes clear. This takes about 30 seconds to 2 minutes. With the tube still on the rack, gently remove supernatant using a pipette. Do not aspirate with vacuum. Remove tubes from the rack and wash beads a second time by repeating steps 2 and 3. Add 10 microliters of concanavalin A bead activation buffer per reaction and gently resuspend by pipetting up and down. If the beads are being prepared before sample collection, activated beads may be stored on ice. Otherwise, proceed to the next step. Visually inspect the beads before combining with cells. If needed, pipette up and down to ensure a uniform suspension. Add 10 microliters of bead suspension per reaction to the washed cell suspension from section 1. Rotate the mixed sample for 5 minutes at room temperature. Alternatively, use a rocker or incubate on a stationary rack. If needed, beads can be resuspended by pipetting up and down. Perform a quick spin to collect any liquid from the cap and sides of tube, then place the tube on the magnetic rack for 30 seconds to 2 minutes until the solution becomes clear. With the tube still in the rack, remove supernatant with a pipette. Remove the tube from the rack and add 100 microliters of antibody-binding buffer per reaction and place on ice. Aliquot 100 microliters of the suspension into separate 1.5 ml tubes for each reaction. Place these tubes along with your cut-and-run and control antibodies on ice. Before adding antibody, pipette the cell bead suspension up and down. Add the appropriate amount of antibody to each reaction and mix by gently pipetting up and down. The amount of antibody for positive and negative controls are shown in this table. Please use the recommended dilution for cut-and-run validated antibodies, which can be found in the datasheet or product page for your CST antibody. Rotate tubes at 4 degrees for 2 hours. This step can be extended to overnight. Alternatively, beads can be incubated on a rocker or simply incubated in a stationary rack for this step before proceeding to the next section. In Section 3, the enzyme Protein A Protein G micrococcal nuclease, or PAG eminase, is added to the cells. The Protein A and Protein G domains recruit the enzyme to antibody bound to chromatin. Before starting this section, check that the digitonin solution is free of precipitates. If needed, heat for 5 to 10 minutes at 90 to 100 degrees before placing on ice. For each reaction, prepare digitonin buffer and PAG eminase premix as indicated in the protocol. Keep PAG eminase enzyme on ice. Briefly centrifuge samples from Section 2 to collect the cell bead suspension from the sides and cap of tubes. Place tubes on the magnetic rack 30 seconds to 2 minutes until the solution is clear. Keep the tubes in the rack and remove supernatant by pipetting. Remove tubes from the rack and add 1 ml of digitonin buffer. Resuspend the beads by gently pipetting up and down, making sure to collect any beads that may be stuck to the sides of the tube. Do not vortex the suspension. Place tubes back on the magnetic rack until the solution is clear, then remove supernatant. Remove tubes from the rack and add 50 microliters PAG eminase premix. Gently resuspend by pipetting up and down. Rotate tubes at 4 degrees for 1 hour to allow the enzyme to bind to antibody complexes. Alternatively, the tubes may be incubated with gentle rocking or a stationary rack instead of rotating. During the 1-hour incubation step, use this time to prepare buffers for DNA digestion, as demonstrated in the next section of the video. At the conclusion of incubation step, proceed immediately to step 1 in Section 4. In Section 4, DNA digestion is controlled by the addition of calcium chloride to activate micrococcal nuclease. If sample normalization is desired for qPCR or NGS, spike-in DNA can be added at this time. During Step 6 of Section 3, binding of PAG imminase enzyme, prepare the buffers required for DNA digestion and diffusion. Check that the digitonin stock is still in solution. If precipitate is observed, rewarm as needed at 90 to 100 degrees. For each reaction, prepare 2.15 digitonin buffer as indicated in the protocol. Place calcium chloride solution on ice. For fixed samples, ensure the 10% SDS is completely in solution, warming at 37 degrees if necessary. For each reaction, prepare 150 microliters of 1x stop buffer as indicated in the protocol. If sample normalization is desired, sample normalization spike-in DNA can be added to the stop buffer.
Refer to the written protocol for guidance on how much sample normalization spike-in DNA should be added for your sample type and analysis method. If both qPCR and NGS analysis will be performed, then prepare duplicate samples with appropriate quantities of spike-in DNA in parallel.
At the conclusion of the 1-hour incubation step at the end of Section 3, briefly centrifuge the samples to collect suspension from the cap and sides of tubes. Place tubes on the magnetic rack for 30 seconds to 2 minutes until the solution is clear, then remove supernatant. Remove the tubes from the rack and resuspend beads in 1 ml digitonin buffer by pipetting up and down. Repeat wash steps 2 and 3 one time. Place the tubes back on the magnetic rack until solution turns clear and remove supernatant. Remove tubes from the rack, resuspend beads in 150 microliters digitonin buffer. Place tubes on ice for 5 minutes. Activate PAG imminase by adding 3 microliters ice-cold calcium chloride to each tube and mix by pipetting up and down. Incubate samples at 4 degrees on a cooling block or in the refrigerator for 30 minutes. We do not recommend performing the digestion step on ice or above 6 degrees Celsius. To each sample, add 150 microliters of 1x stop buffer and mix by pipetting up and down. Incubate tubes at 37 degrees for 10 minutes without shaking. During this step, complexes containing digested chromatin fragments diffuse into the solution. Centrifuge samples at 16,000 Gs for 2 minutes at 4 degrees. Then, place beads on the magnetic rack until the solution is clear. Transfer the supernatant containing enriched cut-and-run DNA fragments to new tubes. If your input samples were fixed, use a SafeLock tube, screw cap tube, or use a tube closure. For live cell or tissue samples that were not fixed, skip the next 2 steps and proceed directly to Step 16. For fixed samples, bring the samples to room temperature, then add SDS and proteinase K to each sample. Vortex to mix, then incubate at 65 degrees at least 2 hours. The incubation may be extended to overnight. This step reverses the cross-linking. After incubation, perform a quick spin at 10,000 Gs for 1 second to collect evaporation from the cap of the tubes. Equilibrate samples to room temperature. At this point, you may proceed directly to Section 5. Alternatively, samples may be stored at -20 degrees for up to a week. In Section 5, input DNA samples are fragmented by sonication. Fragmentation of input DNA is required before downstream analysis of cut-and-run reactions, where the input DNA will be used to calculate signal-to-noise and as a negative control for NGS.
Input DNA is preferred for IgG controls for NGS. Normal IgG antibodies generate low-diversity DNA libraries and can generate what appear to be specific enrichment of genomic regions in cut-and-run, complicating your NGS analysis.
Before starting, thaw the DNA extraction buffer and prepare 200 microliters of DNA extraction buffer for each input sample by adding proteinase K and RNase A as indicated in the protocol. Add to each input sample prepared from Section 1. Mix by pipetting up and down. Incubate at 55 degrees with shaking for 1 hour. Cool samples on ice for 5 minutes. Sonicate samples using a probe sonicator to lyse the cells and fragment chromatin. Incubate samples on ice for 30 seconds in between pulses.
You may need to optimize sonication conditions for your sonicator model by testing different power settings and sonication durations. Scientists at Cell Signaling Technology perform cut-and-run assays and validation testing using an ultrasonic homogenizer sonicator with a 1/8 inch probe on setting 6. Please refer to Appendix B in the written protocol for guidance on optimizing your sonicator. Start with median sonicator power and 4 to 5 pulses, 15 seconds each, with 45 seconds resting time on ice in between pulses. Use 0.5 to 1 microgram of DNA to visualize fragment size distribution on a DNA gel with acidium bromide. The ideal chromatin fragment size range is 100 to 600 base pairs. If you observe chromatin fragments above 1,000 base pair range, modify your sonication program to further fragment the input DNA.
Clarify sonicated lysates by centrifugation at 18,500 Gs for 10 minutes at 4 degrees. Then transfer the supernatant to a new tube. You may either proceed directly to Section 6 for DNA purification or store input sample at -20 for up to a week. In Section 6, cut-and-run DNA fragments are purified prior to subsequent analysis. This section covers DNA purification using spin columns.
We recommend using the DNA purification buffers and spin columns for chip and cut-and-run. These effectively recover DNA fragments 35 base pairs and up, a size range that accounts for over 98% of cut-and-run DNA fragments. If you choose to use other spin columns, ensure the cut-off size is not higher than 35 base pairs. An alternative protocol variation for phenylchromophore extraction and the ethanol precipitation is also included in the full cut-and-run protocol on our website.
Before starting this section, remove one DNA purification collection tube for each enriched chromatin sample or input sample. If you are using a new DNA purification kit, add 24 milliliters of ethanol to the DNA wash buffer, mix well, and mark the bottle to indicate ethanol has been added. Perform a quick spin of your samples, then transfer them to 2 ml tubes. Add 5 volumes of DNA binding buffer to each chromatin and input sample. Mix by pipetting up and down. Transfer 600 microliters to a DNA spin column. Centrifuge for 30 seconds at 18,500 Gs. Briefly remove the spin column from the collection tube and discard the flow-through liquid by dumping into a waste container. Then replace the spin column in the empty collection tube. Repeat steps 2 through 4 to run the entire sample through the spin column. Add DNA wash buffer to the column. Centrifuge to wash the column. Discard the flow-through and replace the spin column in the empty collection tube. Centrifuge one more time to collect all of the liquid from the column. Discard the collection tube and liquid. Move the spin column to a clean tube and add 50 microliters of DNA elution buffer to the center of the spin column. Centrifuge one more time to elute the DNA. Discard the spin columns. At this point, you may either proceed directly to the next section or store purified DNA at -20. Cut-and-run DNA obtained using the cut-and-run assay kit is compatible with downstream qPCR analysis. In Section 7, we will highlight key experimental design considerations for quantitative real-time PCR analysis. Please refer to the written protocol for step-by-step instructions for this section and an example calculation of sample normalization.
The quantity of DNA product obtained after purification is typically much lower compared to other methods for protein enrichment, such as chip, falling well below the detection threshold of instruments such as a bioanalyzer or nanodrug. It is not necessary to quantitate DNA prior to qPCR. Instead, we recommend using real-time qPCR for analysis of a few no-target genes. This acts as a quality control step prior to beginning of an NGS workflow.
Primers included in the Cut and Run kit include those for the yeast ACT1 gene, used to measure signal from the sample normalization spike in yeast DNA, and for the human or mouse RPL30 gene, used to measure signal of the trimethylhistone H3-positive control antibody.
For analysis of other loci, consider the following when designing your primers. For Cut and Run, PCR entry count size should be 60 to 80 base pairs in length. Primers should optimally have a melting temperature around 60 degrees and a GC content around 50%. To confirm the Cut and Run assay is working in your hands, check signal strength and calculate the signal-to-noise ratio for the positive antibody by dividing signal from the positive primer set by the negative primer set. You can also calculate signal-to-noise ratio of your target antibody, also measured at positive and negative primer sets, and the ratio of target antibody to isotype control measured at the same primer set. All three of these ratios should be 3:1 or higher. To normalize qPCR data, use the CT value from the sample normalization primer set to calculate the normalization factor and apply it to the CT values of the Cut and Run samples. Please refer to the Section 7 in the written protocol for details and an example calculation. If your Cut and Run DNA fulfills the quality control criteria described here, you may proceed with NGS.
This concludes the CST Cut and Run protocol. If you have any questions about the Cut and Run protocol that were not addressed in this video or on the Cut and Run resource page, if you need help troubleshooting your experiment, or if you have questions about a CST antibody, please visit cellsignal.com/support. This will connect you directly to the scientists who developed the Cut and Run protocol and who validate Cut and Run antibodies and kits. If you plan to perform NGS analysis of your Cut and Run DNA on the Illumina platform, use the Cell Signaling Technology DNA Library Prep Kit for Illumina systems, validated for ChIP-seq and Cut and Run DNA. Find information at the Cut and Run resource page, or watch the DNA Library Prep Kit companion video next.